Welcome everyone, and thank you for joining us virtually for the latest version of the Variant Effect Seminar Series. My name is Jan. I'm a member of the VEST Organizing Committee, along with Adriani de Souza and Diego Calderon. And we welcome two new members, Mariano Martin and Johanna Bezeres. These seminars are made possible with support and a lot of help from Lara Muffley and Alex Hopkins, and among several others, and our sponsors, the Atlas of Variant Effect Alliance. We're always on the lookout, as a reminder, we're always on the lookout for potential speakers for future seminars. And if there is anyone you would like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I have a few housekeeping items to mention. Each talk is around 20 to 25 minutes, but there should be time for a few questions. Please post those questions at any time on the Zoom Q&A, and we'll read them to the speakers at the end. If you have additional questions, feel free to visit our seminar series Slack channel for more discussions. And finally, we're posting updates on our Twitter accounts, so be sure to follow us there. You can find previous seminars and today's seminar on our CMAP YouTube channel. So don't worry about trying to copy the URL. We'll post them on the chat during the presentation. All the details of the seminar series can be found on our website, including a regularly updated schedule for upcoming speakers. For the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So today's speaker is, the first speaker is Dr. Juannan Zhou. He completed his PhD training in the area of evolutionary biology. He worked in the lab of Mac Cantlich at Cold Spring Harbor Laboratory from 2017 to 2021, with a focus on the development and theoretical computational method for modeling sequence function relationships. He joined the Department of Biology at the University of Florida in the fall of 2021. His current research directions include machine learning methods for modeling genotype-phenotype maps and the genetic architecture of complex trait and experimental evolution. The title of his presentation is Modeling Higher Order Genetic Interaction in Sequence Function Relationship. Juan, whenever you're ready.
Thank you for the introduction, and thank you everyone for coming. Let me share my screen first. Can you see this well? Let me start playing.
Yes.
All right. Okay. So thanks again for coming. So today I'm going to talk about some of my work on modeling higher order genetic interactions in genes and complex traits. So I joined University of Florida in the fall of 2021. So my lab is overall interested in the general research direction of sequence function relationships. And so under this, so we are studying this broad question, by modeling combinatorial demutational scanning data as well as complex traits. In addition to those two topics, which I'll talk about today, my lab's also recently started using yeast to study questions in synthetic biology, using, for example, directed evolution using the so-called orthorep system, as well as high throughput experimental assays. So before I talk about the methods, I would like to thank my collaborators, Carlos, who's going to present in about 25 minutes, and David McCandlish, who's my postdoc advisor, Sam Patty, who recently started her position at Tufts, as well as my co-authors for the first paper I'm going to present, Mandy, Wei Chia, Justin, and Adrian. Okay, so I guess I probably don't have to convince this audience how amazing the MAVE or the DMS technology really is. But in addition to quantifying the effects of all single mutant substitutions, like which is pretty standard nowadays, the MAVE technology is also very good at quantifying the effects of combinations of mutations. So this large combinatorial mutagenesis library provides very important insights about the structure of fitness landscapes, as well as the long-term evolutionary dynamics. And this type of data is also very important for synthetic biology purposes. For example, it can help us design optimal biomolecules such as proteins and antibodies. So, but unfortunately, the DMS experiments cannot take us all the way because the number of possible sequences or genotypes is much more enormous than the physical limit of the experiments we can possibly do. So that means there is almost always a need to reconstruct the full fitness landscape given the experimental data. So the simplest inference method we can build is the additive model. So here we just assume that the effect of individual mutations add up independently. Okay, so that means that if this model is true, all we're going to do is to quantify the effect of all mutations in a given background, and we can just predict the fitness for all possible sequences. So one way to gain some intuition about the structure of fitness landscapes is to plot individual genotypes with their phenotypes on the y-axis and their hemidistance to a given wild-type reference sequence on the x-axis. Okay, like there. And the lines here correspond to the single mutations, right? So if two sequences are connected by a line, that means they differ by at one side by a mutation. So here we have the representation of a simulated additive fitness landscape. And one characteristic of the additive model is that the effect of all single mutations, right? A single mutation is constant in all genetic backgrounds. So here the red lines represent the same mutation, although occurrence of the same mutation, but in different backgrounds. And the slope of those lines then correspond to the effect of those mutations, right? So we see that they're all parallel to each other, meaning that those mutations, they all have the same effect, no matter what kind of background they occur in. All right, so another useful way to examine the geometry of this, the very complex high-dimensional fitness landscape is to look at the mean fitness of all sequences that are a certain distance away from a certain hemidistance away from a reference or the wild type. So here, the red curve is showing you the mean fitness function in reference to this wild type sequence here. So a property of the additive model is that another property of the additive model is that this mean fitness function is always linear. In this case, this linear function connects the global maximum to the global minimum. Okay. All right. So going beyond the additive model, a natural generalization to build more flexible models to fit the data to reconstruct the full fitness landscape is then to fit pairwise models to capture interactions between pairs of mutations. So there has been some recent publications, for example, this paper by Joe Thornton's group that advocate the use of pairwise models together with a nonlinear global epistasis function, since this class of models seem to capture the majority of the variance for certain combinatorial data sets. So here, I want to advise some caution when applying this type of model, the pairwise model, to real data, since this model has some really weird behaviors, as we'll see in a second. So here is a projection of a simulated pairwise model in the same representation as we saw earlier. And here, the red lines correspond to the same mutation, but all of the occurrence in different genetic backgrounds. We see now that the slopes are very different, meaning that the same mutation can have very different different effects when occurring in different backgrounds. So this is a major advantage of the pairwise model over the additive model. Now we can ask the mutation to have different backgrounds by looking at different effects by looking at the background, by looking at other mutations. However, the pairwise model also has other idiosyncrasies. For example, we can examine the interaction among a pair of mutations across all genetic backgrounds in the fitness landscape. So here, all of this like kind of squares correspond to a particular choice of a pair of mutations. And then for any such choice in a given background, we can quantify the interaction between this pair of alleles using this so-called epistatic coefficient. And this coefficient measured the deviation of the double mutant phenotype from the additive prediction, given the mutation effects of mutation of those two alleles. So it turns out that the pairwise interaction coefficient for any pair of mutations is constant in all backgrounds. So although the pairwise model allows mutations to vary across backgrounds, the epistatic coefficient now is constant. And this leads to some kind of weird behavior of the pairwise model. So specifically, if we again plot the mean fitness function against Hamming distance, as we saw earlier with the additive model, it turns out that this function is always quadratic, right? So here is one example where the quadratic curve is convex. And here is one example where the curve is concave. So this means that if we try to fit a fitness landscape with a pairwise model with more expressive forms of distance function, like shown here in the gray curve, where there is a global maximum as some kind of intermediate distance to the wild type, the pairwise model will not do a very good job, even though no matter how much data you give it, right? So here we're giving the model different amount of training data, but through simulated mutagenesis around the wild type, we see that no matter what we do, the model always has this part of the quadratic curve, right? So, so in some cases, this can lead to very inaccurate predictions, like in here, especially for regions that are far away from your data cloud. And the three-way interaction model, where we have like interaction between triples and mutations, also suffer a similar problem with having the same mean function always being, instead of quadratic, is always cubic. So this, what we saw so far is only one diagnostic for those lower order models, but these results show that the pairwise models and also lower order models often make unrealistic assumptions about the geometry of the real fitness landscape, and sometimes may lead to very outlandish predictions. So the first method I'm going to talk about today attempts to generalize the lower order models by including higher order terms into the inference procedure. And with this framework, we aim to build highly flexible models that also have more desirable behavior than the additive and the pairwise and other low order regression methods. So we can motivate our method by considering a simple problem of predicting the phenotype of a triple mutant highlighted in red here. And the very sensible way to predict the phenotype for this particular sequence is then to add to the wild type all the effects of the single mutations, right? As is shown by the arrows here. And we can further make corrections that account for deviation from the additive assumption by adding up the local epistatic coefficients between pairs of mutations, which is our second term here. And then we can make further corrections from deviation from only pairwise interaction assumption by adding those three-way interaction terms. So the challenge in implementing a method like this is that those effects, the individual mutations or pairwise interactions or triple three-way interactions, may not be observed in the data. But the good news is that in a combinatorial library, typically we have many observations for the same effects, but they may occur in different backgrounds, as is shown in this schematic figure for mutational effects. So here, the black arrow corresponds to the focal mutation, the effect of the focal mutation that we want to infer. But we also have the same effects occurring in many different backgrounds that are parallel to this curve, to this line shown here. And same thing for the double mutant interaction episodic coefficient, as well as three-way interaction coefficient shown here. So the question is then, given those observations throughout the sequence space, throughout the fitness landscape, how do we weight them in a sensible way when we make predictions here, right? Because the same effect can appear many different times. How do we weight them in a way that is consistent with the data to make better predictions? Is the question. So here in this paper, we take a data-driven approach So our model works internally by first extracting the correlation or the predictability of mutational effects, as well as this local pairwise high order interactions from the data. So we see that as we move away from the focal, as the divergence in as the background between the backgrounds where the same effects occur become increasingly divergent, the predictability or the correlation in those effects also drops, right? This is the case for the mutational effects as well as high order, pairwise and high order interactions. So our intuition when making this inference procedure is then to take those local mutational and interaction effects and then weight them in a way that is consistent to the observation we have made here. And to see how this method works out, here is one example of its application to a combinatorial data sets of all the 5' splice sites in humans. The figure shows the empirical correlation between the mutational effects as a function of the divergence of genetic backgrounds. We see that this curve drops in a nonlinear way. So our aim is then to come up with a model, or more precisely, a Bayesian method with a prior, where if we draw a sample from this prior, it has the same feature recapitulated. So when we make prediction with this prior, you will correctly weight all of the effects we saw earlier, so we can make a better prediction. So here, I just want to show you that an additive model is not able to do that because the correlation of mutational effects is constant, is always 1. And that is just the assumption of the additive model, that the effect of a mutation is always the same, no matter which background it occurs. So that additive model cannot capture this pattern we see in the data. And neither can the pairwise model, because it turns out that the distance correlation of mutational effects of pairwise model decays linearly, which also strongly deviates from the nonlinear pattern we see here. So the trick here is then to introduce higher order interaction terms and tweak their contribution to fit this curve. And we do this by inferring the contribution from different orders of interaction with this quantity we call the variance components. So by tweaking the variance components with the contribution from different orders of interaction, we can recapitulate this, this decay or this correlation in the effect of mutational effects in our prior. We can then incorporate this information into a Bayesian inference framework. And when we do this, we see that we achieve a much higher predictive performance than the pairwise model and even the three-way model. So here on the right, I'm showing you the out of sample R squared of our method under different amount of training data against the three-way and the pairwise model, as well as the global epistasis model shown here. So one drawback of the method I just mentioned is that it assumes that all sites and mutations contribute equally to determine fitness. But we know that this assumption is often not true. For example, in some cases, some nucleotides on the 5' splice site are more important than others. So this motivates us to work out a generalization of the first method. To do this, we introduce this site-wise site-specific factors, what we call rho here, such that the contribution from the interaction among 3 sites, for example, are proportional to the product of those site-specific parameters rho. So this gives us a model where we can still model higher order interactions, but the contribution from different sites can now be tuned more independently than before, which allows us to fit more detailed features in the data and to allow better prediction. And this model also has some strong theoretical connections to the so-called connected model proposed by Gautam Reddy and Michael Desai about 2 years ago. So here on this slide, I'm showing you the application of this method to the same 5' splice site. And the top left panel shows the site-specific contribution, which is the rho parameter. So the higher the magnitude is, so the more important this site is. So the more strongly it's contribute to the single mutant effects as well as when it occurs in an interaction. Well, the bottom panel shows the site-specific decay factor. As this sites are mutated, we see that the more important sites on the top panel, when mutated, results in a faster decay in the phenotypic correlation. And in terms of model performance, we see that the row kernel now, or the connectedness regression, shows a substantial performance improvement over our old method, which confirms our suspicion that the nucleotides on the 5' splice sites, they vary greatly in their importance. I guess I'm doing good in time. So for the last part of the talk, I will talk about a generalization of the same modeling philosophy, but moving away from working with DMS datasets to studying complex traits for diploid organisms. So studying complex traits for high organisms, especially in particular diploid organisms, pose some very serious challenges. For example, in diploid genomes, interactions among alleles cannot just occur among sites, but also within the same site, which is dominance, right? And to make matters worse, there can be a full spectrum of dominance by epistatic interaction. And this causes the geometry of the fitness landscape for diploid organisms to be very, very complicated, a lot more complicated than we can possibly have for like haploid genomes, such as genes and proteins. So here I developed a method that is able to model the full spectrum of those interactions we saw earlier in this slide, which is completed with the efficient parameterization of the large number of various components and GPU acceleration enabled inference. So I first applied this method to the large diploid barcode East library constructed by Sasha Levy and Ian Arorek's lab. So here they have 100,000 diploid strains of yeast resulted from the mating of two parental strains. And because all of the strains are barcoded, we can simply grow them in different conditions and sequence the genomic barcode to get the condition-specific or the environment-specific growth rate and do the same thing as we've done earlier. Basically, take the data, fit a model, and make the prediction and see how well our model does. So so on the right, I have the performance comparison between our method and the additive model on the bottom, as well as a commonly used standard nonlinear regression model using the so-called Gaussian kernel. We see that our method achieves a much better prediction accuracy than the other two other models, which means that that in this landscape there is more complicated patterns of genetic interaction that can be captured by this nonlinear model shown in the middle. So this theory also provides us with a framework. In addition to making accurate predictions, this theory also provides us with a framework to interpret the posterior distribution in order to investigate the genetic architecture of the genotype-phenotype maps. So technically, this method is centered around a set of tricks to perform various component analysis for single sites, pairs of sites, or set of genes, etc. So in a way, that is similar to analysis of variance or ANOVA type of methods. So here I'm showing you one application of this framework to the problem of identifying strong pairwise interactions among the large number of SNPs. So here the challenge is that we have now, like, instead of having only hundreds of sites, for this type of like large East crosses or human observational data, there are usually like a very large number of segregating sites, usually on the order of thousands up to millions of sites. So the challenge here is that the number of pairwise tests that scales quadratically with the number of SNPs, so that in this specific East data set, we have 8,000 SNPs, which means we need about 3 million pairwise tests, which is just a little bit too much. And if we are working with human data sets where we have routinely millions of SNPs, then we have like hundreds of billions of tests to do. So the question is then how do we reduce this computational burden? So our solution here is then to come up with a way to prioritize only a subset of all the SNPs. So the way to do this is to estimate the marginal epistatic effects for all the loci we have in the data, which is equal to the variance explained by all the interactions involving a focal locus. So after we have quantified those marginal epistatic effects and identify loci that are typically involved in large numbers of interactions, we can then only prioritize those loci with large effects for downstream pairwise testing. So on the top panel, I'm showing you the marginal effects of all the SNPs across the yeast genome. We see that there are some of those so-called epistatic hubs, which are involved in large and also strong epistatic interactions. So then we can just draw a line. For example, we can pick the top 100 of those SNPs and only perform pairwise testing between those SNPs and everything else. So after we have identified those pairwise pairs of SNPs to test for strength of interaction, we then came up with a method to quantify pure pairwise interaction that is free from the background influence by simply projecting the posterior to the space of pure pairwise interactions. So on the right, I'm showing you the interaction map among all the 8,000 SNPs in this data set. So the question now is then, after we have this interaction map, how do we extract important like interpretable biological information insight from this? So another question is related question is then, given this data, can we extract important or high priority gene interactions or pathways? So another thing I'm working on is to, in the same using the same philosophy, is then to, instead of quantify the variance explained by single genes or single SNPs or pairs of SNPs, we can also quantify the variance explained by the set of genes, for example, like genes in a pathway. And then we can do this for all the candidate pathways and identify pathways that are important for contributing to the variance in fitness for certain growth conditions. And then do some kind of forward genetics and to isolate those pathways for downstream analysis. So to conclude my talk, in the beginning, I talk about how lower order interaction models often assume very specific types of geometry. And if we use those models for inference, sometimes this can lead to unexpected and unrealistic predictions. And also make the case that for high order genetic interactions, which is good for allowing much higher flexibility to capture the background dependency of mutational and local interactions. And we see that when we do this, we do see a substantial improvement in model performance. And specifically, I'll talk about two methods for modeling mutational scanning data. The first one works by picking the mixture of orders of interaction that matches how phenotypic predictability decays with the background, while the second method, I generalize the first method by inferring the multiplicative site-specific decay factors, which can further improve upon the first method. And I also talk about the methods for modeling complex traits, where we're able to capture the full spectrum of epistatic dominant as well as mixed type of interactions. And this method is also completed with analysis of variance type of techniques for studying the genetic architecture of the complex traits. So with this, I would like to thank my collaborators, Carlos, David, and Sam, and also thank you for your time. And thanks for listening. I'm stop sharing.
Thank you. Yeah. Thank you, Juannan, for your great presentation.
Thank you.
While we wait for more questions to arrive in the chat, I have some. Let me check if you have one already. No? Okay. So our first question is, if someone has experimentally tested the fitness landscape in different genetic backgrounds in a traditional MAVE study, can your model integrate those measurements considering the Hamming distancing from wild type? Would it change the model?
Sorry, can you read the last part of the question again?
If someone has experimentally tested the fitness landscape in different genetic backgrounds, can your model integrate those measurements, considering the Hamming distancing?
Yes, of course. So what our model is good at is picking up those background dependencies, right? So I think what the audience or what the person meant was, if you do the position weight matrix type of analysis across different backgrounds, can our model integrate these measurements into a coherent inference procedure? So I think the answer is yes. The answer is yes, and then we can see how backgrounds. But what's gonna make the model more so the answer is yes, but what will make the model more powerful is if we also have like triples or double of mutations.
Okay. It was actually my question because I have
That's a
tested
good
a
question.
triple
Yeah,
mutant.
because those type of data, they are becoming more available, right? But we haven't really applied our method to this type of data, which will be very interesting.
Oh yeah, great.
Yeah, that's the question.
We can catch up. Okay, we have another one. So do you find that the wild type genotype is the most fit? And as you add more mutations, do you typically decrease in fitness? Does this have to do maybe with how yeast, as an example, is adapted to the lab environment and more mutations, of course?
That's a great question. So that is definitely not always the case. And the way I was talking about the wild type is because it was only a way for us to look intuitively at the structure of the geometry of the fitness landscapes. But there are also different projections where we have the wild type or any kind of reference sequence not being the most fit sequence. But the bottom line is, if you fit a Paris model, it'll always have this characteristic quadratic curve with a mean fitness function. But our model does not assume like, the wild type is the best model. That is the assumption made by something like the additive model or the global epistasis model, where there's always a global maximum. But our model actually allows like multiple fitness peaks to appear, right? To occur in the fitness landscape. But it's also very good at, if the landscape is simple, we can do like a single peak landscape. If the landscape has many, many peaks, we can do that as well. Yeah, so the answer is like we do not require like a single peak landscape, and the wild type does not have to be. Yeah. And I believe there are cases where like, for example, in GIP, I need to check my facts, but I think in some of the Contrashop papers, some of the like the mutant GIP has like much better activity than the wild type. Yeah, thanks for the question.
Let me see if we have more questions for people attending. No. So I have one more question related to the last topic of our presentation about higher order genetic interaction. I'm wondering if there's a limit for the amount of genes you can analyze with your model using higher order genetic interactions. As an example, I know of the gene family 1433 that have 7 genes in the family that is known to have higher order genetic interaction in plants, but not in mammals because it's hard to study.
So that is a really good question. So short answer is computationally, there isn't a limit to the number of genes because our model doesn't work. The complexity does not scale with the number of genes or even the number of markers. But there is something that is slightly tricky, which is if you have a lot of genes, the genes carry like the number of SNPs increases. So we have to come up with some kind of null distribution. Basically, you have to control for false positives. So, but computationally, there's no challenge. The challenge comes from the multiple testing side. Yeah, I hope that makes sense. Yeah, I'm happy to talk more in detail about this issue, which is very interesting. We just recently started working on this direction.
I think you explained well. Thank you so much for presentation and answering the questions. And I think if we don't have more questions, we can move on to the next presentation.